Good morning, students. Today we are going to learn about the technology which is called as gel electrophoresis. The main principle behind this technology is the migration and separation of the charged particles under the influence of an electric field. And the material required: plastic box with wells, agarose gel, sodium bicarbonate, battery, electrodes, connecting wires, and loading sample. So this is how we set our setup for the gel electrophoresis. In this part you can just see that we just have a rectangular box with the wells where we can going to load our sample. We just using our sample, loading sample. Maybe it would be the DNA sample or something else. The battery eliminator, which we going to set for 9 volt to 12 volts, and connecting wires, electricity supply. Now we are going to load the sample into the wells. Like in this portion of this video, you can just see that I loaded the sample, that is I am going to use the two dyes, that is methylene blue and saffronin, and I load these dyes into the wells of the rectangular chamber, and I set up the apparatus to the battery eliminator and using the current at 9 volt to 12 volt of it. Now you can see in this portion of video the beautifully my sample is going to run on this agarose gel. I used in this rectangular box the agarose gel. I am making the bed of agarose gel, and then beta we are going to use the buffer solution which is made up of sodium bicarbonate. And you can just see that how beautifully the gel, the loaded sample, is loaded onto the agarose gel under the influence of electric field. You just see, and it is very clear. The lighter things are to be moved faster and the heavier particles are moved slowly. In this way, the gel on the gel bed, the sample is loaded up and the sample is traveled from one terminal to the another terminal of it. So this is the basic principle of the gel electrophoresis, that is the separation of the ions under the influence of the electric field, and it will go from one terminal of the battery towards the another terminal of the battery. So the gel is start moving on to the sample is just start moving on to the agarose gel based medium. Now in this portion of this video you just clearly show how it can be travel after 10 minutes, then 20 minutes, and then after to the 30 minutes. So beta, if on the contrary of it we are going to use the DNA samples on it, so it will start moving from one terminal towards the another terminal in the phase of 30 minutes, and in this way we can get a beautiful band patterns after giving them to stain with the ethidium bromide. So application of this, it is used in DNA fingerprinting, PCR, DNA profiling, paternity testing, and evolutionary